next talk that's going to be by Cecilia Vranych. She will talk about how cannabinoids mobilize cholesterol in C. elegans through the insulin pathway.
Well, good morning, everyone. My name is Cecilia, and I'm going to share with you some results about our recent work denominated cannabinoid system. Cannabinoid activate the insulin pathway to modulate mobilization of cholesterol in C. elegans. Although cannabis extracts have been used in medicine for centuries, the past few years have seen an increased interest in the medicinal use of cannabinoids for treatment of many diseases in the nervous system. However, the endocannabinoids like 2-arachidyl-glycerol or 2-AG and anandamide are produced naturally by the human body. The endocannabinoid system in mammals is a molecular system of communication which includes receptors, enzymes, and endogenous lipid ligands, the endocannabinoids, that trigger intracellular signals via second messengers, and with an important roles in several processes such as homeostasis regulation, neuromodulation, memory learning, and others. In C. elegans, the endocannabinoids are present, like 2-AG and anandamide, and place multiple effects in longevity, axon regeneration, movement, aversive behavior, and it was shown by our group some time ago. The endocannabinoids in C. elegans are essential for the mobilization of cholesterol from internal reserves, a process or critical process to maintain the integrity and function of the cells and to promote the reproductive development. In this way, by assays under cholesterol depletions, a growing condition, we could observe that C. elegans arrest to dower-like larvae in the second generation without cholesterol. And 2-AG is able to rescue C. elegans from this phenotype induced by the hunger for cholesterol, indicating that 2-AG or the endocannabinoids act as messengers to stimulate the mobilization of cholesterol to promote the production of dafachronic acid, essential to trigger the reproductive development by DAF12. This process is a consequence of the several pathways, including DAF7 pathway and DAF2 pathway, or the insulin pathway, that modulate the conversion of cholesterol or to cholesterol into dafachronic acid. Under unfavorable conditions, growing conditions, these pathways are turned off and the dower problem is promoted. So in order to decipher or elucidate the mechanisms by which the endocannabinoids rescue the C. elegans from under cholesterol depletion, our main question was, which are the signaling pathway mediated by endocannabinoids that promote cholesterol mobilization in C. elegans? Initial reports suggested that C. elegans lack the canonical CV receptors, but possess cannabinoid-like receptor, naming NPR19 and NPR32, with conservation of the critical amino acid residues involved in the endocannabinoids binding. In order to observe or analyze if these receptors are involved in sense of 2-AG, we perform assays under cholesterol depletion, growing condition, and using mutants in these genes. And we could observe that, similar to the control, these animals arrest to dower-like larvae in the second generation without cholesterol. And then when we add 2-AG, we could observe that 2-AG relieves the larval arrest in all mutants, including the double mutant. Where can you see? So this suggests that 2-AG modulate cholesterol homeostasis through NPR 19 or 32 independent pathway. In mammals, many cannabinoids can activate TRPV channels. One group of transmembrane receptor included within the TRP receptors be able to form channels for ions in trans with preference for sodium and calcium. And in this way, 𝐶𝑎𝑒𝑛𝑠, the 𝐶𝑎𝑒𝑛𝑠 genome encode for 5 members of this group of transmembrane receptors, denominated OCR1, 2, 3, 4, and OSM9. And in order to observe if these proteins are involved in the signaling pathway mediated by 2-AG to promote cholesterol mobilization, we perform assay under depletion, cholesterol depletion, and using these mutants in these receptors. And we could observe that in contrast to OCR1, 3, and 4 that produce almost 100% of gravids adult in the first generation without cholesterol, OCR2 and OSM9 produce animals display a high incidence of arrested downward light in the first generation without cholesterol. And the 2-AG was enabled to rescue this arrest, suggesting that OSM9 and OCR2 TRPV channels could be essential for 2-AG dependent cholesterol mobilization. Also, OSM9 and OCR2 not respond directly to 2-AG, as was demonstrated by in vivo and in vitro studies, suggested that it could be acting upstream of TRPV channels in sensory transduction. Previous reports have shown that the temperature-sensitive DAF2 and DAF7 mutants are also sensitive or hypersensitive to depletion of cholesterol under depletion condition, forming downward at already 20 degrees. But when we added the 2-AG, we could observe a partial rescue of the animals that expressed DAF7 mutant, suggesting that the cholesterol mobilization by 2-AG is independent of this pathway. However, 2-AG couldn't suppress the downward arrest of the DAF2 mutants, indicating that an active DAF2 or insulin pathway is needed to the mobilization, the cholesterol mobilization by 2-AG. The insulin pathway has several physiological functions in the worm, and one of them, the activation of the FOXO transcription factor DAF16 by a phosphorylation cascade. In this way, when DAF16 is not phosphorylated, is active and translocate to the nucleus to promote the arrestal larva. In other words, when DAF2 is turned off, DAF16 is active and DAF16 is in the nucleus. So according to this report, we perform assay under depletion, cholesterol depletion, growing condition, and using a strain that express DAF16 GFP fusion, and we could observe that after a second generation without cholesterol, these animals exhibit a high accumulating a high accumulating of the fusion protein, the protein fusion in the nucleus compared to the control worm grow in the supplemented medium with cholesterol. In contrast of this, the exogenous 2-AG largely inhibit the nuclear localization of DAF16, reinforcing the hypothesis that the endocannabinoids are essential to mobilize cholesterol and induce the production of dafachronic acid. So these results show that the insulin pathway and 2-AG converge in a process essential for cholesterol mobilization. As a conclusion, we could observe that the 2-AG or the endocannabinoids are essential to mobilize to the transport and cholesterol mobilization, and this pathway is independent of the known cannabinoid receptors NPR. And we propose a working model where you can see a dual role of the 2-AG by one, firstly, to remove the cholesterol from internal pools by interaction to promote the intracellular cholesterol trafficking, and then enhancing the production of the dafachronic acid that bound to DAF12 to promote reproductive development. On the other hand, 2-AG by a mechanism as molecular specific activate the OCM9, OCR2 TRPV channels that stimulate the production of insulin peptides that stimulate the DAF2 or insulin pathway that together the dafachronic acid bound DAF12 avoid the DAF16 nuclear translocation, and in consequence, the larval arrest in C. elegans. Also, the upregulated of the regulation of DAF2 produce energy to provide energy to the intracellular cholesterol mobilization, and other mechanisms involves the SVP protein. At last, we're finished. We would to thank my dear lab, and especially Bruno Hernández Cravero that performed the assay, and the work team, and our collaborators. Thank you for your attention.
Thanks, really nice talk. So we work both DAF7 and DAF2 DAWers in the lab, and while they look similar, they are totally different. I mean, we can rescue many mutations with DAF7, but with DAF2, we say always that it's a really strong allele. So do you think, based on the cholesterol mobilizations data that you showed, that if you can measure the dafachronic acid in both DAF7 and DAF2, I mean, it will be totally different, the amount? I mean, you could expect to have more dafachronic acid in the DAF2?
It's a good question, but I don't know. I don't know.
Thank you very much, Cecilia.
Thank you.